the carbohydrate fermentation media. You want to make sure to have your handout available, which should be available on Canvas, which has all the information that you need to fill out for your handout. It's very important to work on getting your handout completely filled out, otherwise you're going to have a bunch of gaps, and it's a very good, useful study guide, so make sure you guys fill that out as you go along. And bring it with you during things like when we do unknowns or various types of tests. It's a great study reference for the midterm and the final. So make sure you guys fill it out, keep it with you, keep it in your lab book, something like that. All right, for carbohydrate fermentation lab, a lot of pictures are in here. Some really great resources is your photographic atlas that has a lot of great pictures. You can also look in your appendix 5 for some information on those, but the atlas has really great pictures and examples of each type of media in case you happen to forget what it looks like. Okay, objectives and safety stuff. All your objectives are on page 117. The quiz will cover sugar fermentation lab. There will be a lab report due at the end of lab as usual. Some safety stuff to consider is that we will be using some BSL-2 organisms. Make sure you guys follow proper safety protocols as usual. Things like making sure you have proper protective equipment, goggles, gloves, and lab coat, long pants, closed-toe shoes. Make sure you don't drop anything. Try to avoid that. Notify your instructor of any breaks or spills. Okay, so the purpose of the carbohydrate test. We want to differentiate organisms into 3 separate categories, whether they're able to ferment a sugar, oxidize a sugar, or non-saccharolytic or asaccharolytic towards that sugar. So if they're able to ferment, that means they're able to break it down without the presence of oxygen. If they're oxidative, they need oxygen to break it down, so they burn it. They have systems similar to what we have, where we burn the sugars with oxygen. And then there are organisms that cannot utilize that sugar at all, and they're called non-saccharolytic or asaccharolytic. Either word is fine, but they are both of they just can't use it at all. To determine the specific sugars that can be fermented into acid with or without the production of carbon dioxide is the other purpose. So we're basically saying, okay, if you can break down the sugar, can you ferment it? If you can ferment it, we know that you're going to produce acid or carbon dioxide. And there are 3 main byproducts of fermentation. You do need to know them. They're acid, usually from lactic acid. Occasionally this isn't always true, but by and large it's true. So you'll get an acid product, you'll get alcohol, about fermenting, you'd get alcohol as a common byproduct, and last but not least is CO2. Now alcohol, pretty hard to test for, not really worth it, but acid and CO2 are really easy to test for, so we use those quite a bit. Fermentation can happen in the absence of oxygen, so that's important to remember. So if you're an oxygen organism, if you're oxidative, you cannot ferment because you cannot survive without oxygen, so that just doesn't happen for you. A couple of quick things to note. Whenever you guys record tests, specifically for this lab, this type of answer thing, anytime you record one of these tests, you need to record each of the letters listed in your handout. So it'll have like a bolded letter A, B, and a C. Make sure you record all those points, otherwise you're going to lose out when you do your unknowns, for example, or any other type of test where you have to respond to that. So, for example, a TSI slant has 3 results for it. You need to make sure to have all 3, or else you get a 0 for those. So make sure you guys remember to put those in. All right, first one up is the phenol red broth with Durham tubes. The pH indicator in here is phenol red, and for each of these, I'm going to list off what the indicator is, so make sure you guys write that down. So in this case, it's phenol red, which is very commonly used. Generally, it sits at a red color, so that means it's alkaline. So we normally make the media so it stays at a red color. If it turns to orange, it means it's a neutral solution. But for this particular test, it doesn't really matter if it gets to orange coloration. We don't really care about that. We really only care if it changes to yellow, which means acid production. If acid's being produced in high amounts, that indicates that fermentation is going on. So we would say that this is really important. Now, the sugars that we're going to be utilizing are glucose, also called dextrose, so that's the dimer of that, or lactose or sucrose. So make sure you guys remember that each tube has its own type of sugar associated with it, and that'll usually be labeled on the tube itself. So make sure you guys keep an eye out for that. Okay, phenol red sugar broths with Durham tubes. The Durham tube is this little tiny test tube inside of here. You can see it. The purpose of this test tube is to trap a little CO2 bubble in there. One of the key things when inoculating these, I like to use loops, but you can also use a needle. You want to make sure to give it a good swirl in here and try to get underneath at the bottom of where this tube sits so that the bacteria can get in here so the CO2 can easily be trapped. Otherwise, it kind of creates a seal down here. The reactions can have three possible results. The first one is alkaline with no CO2 bubble, so that means the bacteria was either unable to utilize the sugar, or is oxidative, so it cannot break down fermentatively, so it just stays red like this. And then there'll be no CO2 bubble because, of course, there's no fermentation going on. You can also be a fermenter, so you produce acid, you're able to utilize the sugar as a fermenter, and you might not be able to produce any CO2. So sometimes CO2 is produced at such small amounts that the bacteria just don't produce enough to measure, so we just can't really see it. So we just say that that's acid with no CO2. The last one is acid with CO2, and you can see a CO2 bubble trapped right here. So you can see it's yellow broth, it means acid production, and then that CO2 bubble is trapped inside that Durham tube right there. It's actually raised the level of the liquid, so you can see that it's pushed it out and raised up the level on the outside. And that would indicate that it is a fermenter with CO2 positive. So we'd say it's positive for whatever sugar it is. In this case, you can see right here this little L. That means that this is a lactose broth. So it's say it's positive for lactose fermentation, CO2 positive. Now the next type of tube we use is OF glucose deeps. These are green semi-solids. OF stands for oxidative fermentative, so it's OF media. It has glucose as the main sugar in there. The indicator is bromthiamyl blue. This media is interesting in that it's always used as a pair. So if you don't have a pair, it is basically worthless. So make sure you guys always grab two of these tubes whenever you use them. We call them open and closed, but that actually doesn't mean like the caps are off. What that means is that they have either a layer of oil or no oil. So if it's got a layer of oil, it closes off the bacteria from any oxygen sources. If it has no oil, we call it open. That means that it's open to the oxygen atmosphere and can receive oxygen. There are three possible results. You can either be a fermenter, which typically means that you're in the Enterobacteriaceae family, or you're able to oxidize glucose, so you're an oxidative organism, or you're non-saccharolytic. You cannot utilize glucose. There's very few that fall into this category, but there are some bacteria that do. So you got these three results. Now, normally the media is in these green settings. So usually green just means nothing's happening. It's neutral pH, nothing's occurred. And so if you have a green tube, that means the bacteria was not able to utilize the sugar at all. It couldn't utilize it in oxygen or in the absence of oxygen, so this is just non-saccharolytic. That's how to record this. Now, if it was able to utilize the sugar in the presence of oxygen, but not in the absence of oxygen, we would have a yellow-green tubes. So the yellow tube that's open to the air means that it's able to break it down with oxygen present. If you take away the oxygen, the bacteria cannot utilize it, so that means it's oxidative. It has to have that oxygen to break down the sugar. Last one is glucose as a fermenter. You can see that it breaks it down in the presence of oxygen or in the absence of oxygen. It doesn't need oxygen to break down the glucose, so that means it's a fermenter. It can break it down in the absence of oxygen. So yellow means that's that acid being produced and shifting that pH down. Remember how we talked about green being neutral, so that means relatively nothing's happening if you have a green color. There's one last possible result, which is blue coloration, and a lot of times you'll see it in the green tubes, especially when it's got open to the atmosphere. It'll turn maybe a little blue on the top here. Blue means that it's an alkaline byproduct. This isn't really significant in terms of something that we'd record in the OF glucose steeps, but it should be something you're aware of. It means that the bacteria is starting to utilize the proteins in here as an energy source because it's just running out of nutrients. So it starts using the proteins, and that creates alkaline byproducts and raises pH up to that blue color. Thus the name bromthiamyl blue. All right, the last of your tubes is the triple sugar iron slant. This auger slant contains three types of sugars, a very tiny amount of glucose, just a little bit, and also some lactose and some sucrose. The pH indicator in here is phenol red. And we also have a secondary indicator that's not a pH indicator. It's called ferrous sulfate. It lets us detect the production of hydrogen sulfide. So if hydrogen sulfide is being produced, it'll react with the ferrous sulfate and create this black precipitate. There are three separate results you should always record. You should record the presence of absence of acid. You should record the presence or absence of carbon dioxide and the presence or absence of hydrogen sulfide. Acid products will create a yellow color on the auger. So this, if you guys remember, we always set it to the red, so the alkaline product would be red, so that means nothing's happened. Neutral orange color doesn't really mean anything for us, so we don't really care about that. But the yellow color means acid's being produced because it's fermenting that sugar. Carbon dioxide, in this case, looks like bubbles in the agar or cracks. Sometimes it'll break the agar apart or kind of push it up out of the tube, so keep a sharp eye out for that. And last one is hydrogen sulfide, which is black precipitate. This is black precipitate. You can see right here, it's very, very dark. It'll actually block out the light. Occasionally, what will sometimes happen is that the tubes, especially tubes like this one that don't really have anything growing on them, will have a little bit of black grittiness sinking down to the bottom. That's just the ferrous sulfate coming out of solution. It's not, it doesn't indicate anything. It's just stuff that's coming out of solution. So it has to be something a lot like this where it blocks out all the light. So the first result we can have is this one right here. We inoculate it. It is not able to utilize either sugar as a fermenter, so it's probably indicates that it's a oxidative organism. So we record this as K/K, which is alkaline over alkaline. So it's got an alkaline slant and an alkaline butt. So this is the butt of the tube. This is the slant. So we'd record K/K. And remember, we always record 3 results. So we'd say K/K, CO2 negative, because it cannot ferment, it cannot produce CO2. And then we'd record its H2S negative because there's no hydrogen sulfide. Here we can see that it's red on top, yellow on bottom. So we record this as K/A, alkaline over acid. We can see the CO2 bubble here on the side and at the bottom over here. So we'd say this is CO2 positive and H2S negative because you don't see any black precipitate. This one over here you can see is yellow on top, yellow on bottom. And that means that it's alkaline over, acid over acid, so A/A. You can see the CO2 bubble over here, and you can see that there's no black precipitate. And that no black precipitate indicates that it is H2S negative. The last one we can see is A/A. Now, even though it's really hard to see the bottom over here, what we can assume is that if it gets the top slant over here, that it's also going to get the bottom. So if you're able to ferment anything, you should be able to ferment glucose. And what the bottom of the tube indicates, so when we've got the bottom active in all 3 of these tubes, that indicates that glucose is being fermented. So that's kind of the first hurdle there. We say, okay, if it's going to be fermenting anything, it's going to ferment glucose, which is the reason why we only put a little tiny amount of glucose in the media. Then we do the slant, and the slant tells us if it can ferment either lactose or sucrose. We don't know which one. It's one of those, but we don't know which one for sure. So we'd have to do further tests to find out. So if it's going to ferment anything, it's going to ferment glucose. So even though it's really hard to see here because of the black precipitate, we do know that it's A/A, so we know it ferments glucose and at least one other sugar. And then we've got the black precipitate here, so it's H2S positive, and we can see this little tiny CO2 bubble. You got to keep a sharp eye out for those because sometimes it will kind of be hiding there, and so you have to keep an eye out for those kinds of things. All right, for the back side of the paper, we have the mannitol salt agar plates. These are really bright red when you look at the side of the plate, and that's usually how you want to look at the plates when you're trying to analyze what color they are. Just to differentiate them is if you look at the side, it's a lot easier. The pH indicator in here is our old friend phenol red. The sugar that we're looking at is mannitol, and it contains 7.5% salt, so that's a very salty media. It inhibits anything that doesn't like to grow in salty environments. Whenever we talk about plates, in this context, we're talking about selective and differential plates. So selective means I only allow a certain type of bacteria to grow on me, and differential differentiates between some property of those bacteria growing on the plate. So in this case, this plate selects for gram-positive halophiles. So if you have to be both of these things. You can't just be gram-positive. If you're gram-positive, like say Lactococcus lactis, that's gram-positive but not a halophile. So it will not grow on this plate at all. You have to be a gram-positive halophile, so that means a salt lover. And if you're not both of these things, you're just not going to grow. So if it does grow, it's a gram-positive halophile. If it doesn't grow, it's not a gram-positive halophile. It could be gram-negative. It could be gram-positive but not a halophile. So you got to keep that in mind. So make sure the difference between these two is gram-positive halophile or not a gram-positive halophile. And then from those, you can differentiate those who are able to ferment mannitol from those who cannot. If you're able to ferment mannitol, you create a yellow, bright yellow zone on the agar. If you stay pink, then you will be mannitol negative. You cannot ferment mannitol. So here you guys can see we've got a dark growth line here and a growth line here. So these are both gram-positive halophiles. This one, however, didn't change the color of the agar, so that is mannitol negative. So we'd say it's a gram-positive halophile, mannitol negative. Here you can see it change it to a bright yellow, so we'd say gram-positive halophile, mannitol positive. It can ferment mannitol. The next plate is McConkey's agar plate. It's got kind of this violet or faded red look when you look at it. It has the sugar lactose in it. It's got these bile salts and neutral red and crystal violet in there, which help prohibit growth of gram-positive microbes. The indicator in here is neutral red and crystal violet to a greater extent. So these two basically help give you the indicators in this. It selects for gram-negative organisms. So that means that it's really good at preventing gram-positive growth. So gram-positives just cannot grow on there at all. So if you have bacteria growing on there, you can say, yes, they are gram-negatives. It's differential for lactose fermentation. So it differentiates between weak and strong lactose fermenters. So you could either be a non-fermenter, you couldn't ferment at all, but still grow on there. The bacteria would just kind of be this tan bacteria color. The agar would be kind of tan. It wouldn't really have a color to it. If you're a weak fermenter, the bacteria are going to have this pink coloration, but the agar won't really have any coloration to it. If, however, you're a strong fermenter, you will have a hot pink colonies with this hot pink halo around it on the agar. So what that looks like is this. This right here is a strong lactose fermenter. So you can see growth on here. We'd say, yes, this is a gram-negative organism. And you can see this hot pink colony, so the bacteria, this hot pink color, and it's got this halo around it. So we'd say, yes, this is a strong lactose fermenter. If, however, the bacteria just kind of take on this purple pinkish color, we'd say, okay, that is a gram-negative organism, and it is a weak lactose fermenter. And here you can see the example of the non-lactose fermenter. It just stays kind of like this bacteria color. It doesn't really have a coloration to it. So we just say that this is a non-fermenter. It can't really do anything with lactose. And you can see right next to it some strong lactose fermenters with that halo around it. And if you're really talented, you can actually do some art with it, which is pretty cool. But yeah, you should remember that. Strong lactose fermenter, pink colonies, pink halo. Weak lactose fermenters, just slight pink coloration. Non-lactose fermenters, tan, no coloration of the agar. Now the last plate we're going to talk about is eosine methylene blue, or EMB agar plates. These are very dark maroon colors. They're the darkest of the three plates. The sugar in there is also lactose, and the indicators are eosine Y and methylene blue. They inhibit the growth of some gram-positive microbes. So that's the key thing right there, that's some. So it should select for gram-negatives, and some gram-positives will grow very weakly on there. So if you have a very aggressive gram-positive, it'll grow on there. So it's not a very good selective medium, but what it's really, really good at is differentiation between lactose fermenters. It's really easy to differentiate strong and weak lactose fermenters. Now you might be saying to yourself, but Josh, we just did a plate that did selection for gram-negatives and lactose fermentation. Well, they actually do two different things. They do one thing better than the other one. So eosine methylene blue, very good at differentiating lactose fermenters. There's not a lot of overlap in color, whereas for the McConkey's agar, it's much better at selecting for only gram-negative organisms. So this plate isn't very good at selecting, but it's very good at differentiating. McConkey is very good at selecting, but not very great at differentiating. So EMB plates, you can either be a non-fermenter. If you're a non-fermenter, you'll just stay kind of translucent. You don't really have a coloration to you. If you're a weak fermenter, you'll kind of take on this dark maroon coloration to the colonies. Just you'll pick up a little bit of dye, but not too much, and so you'll just kind of have a dark coloration to you. If you're a strong fermenter, though, you will have a metallic green sheen on you. So this metallic green sheen indicates that you are a strong lactose fermenter. You can see an example right here. This is usually a good test for E. coli, which is the organism on here right now. You can see it's this shiny green. That means that it's yes, really easy to identify as a strong lactose fermenter. You can see the other three results. This is a gram-positive organism, doesn't grow on here at all. A gram-negative, but non-lactose fermenter, so you can see it just kind of has that translucent bacteria color, doesn't really look like anything. A weak fermenter is kind of this dark maroon color, it's got that pink pigment. And lastly is the metallic green sheen you can see right there. Pretty easy to differentiate. Make sure that you're paying attention to these. These are really good study guides, so make sure you fill out that handout and let us know if you have any questions. Thank you.